Hello, everyone. We're going to continue today with the material that's covered in Module 4. And today we're going to talk about genetics and how we inherit traits from our parents. So last time we talked about meiosis, which is the process for producing sperm and egg cells. So they're each haploid. They have one set of genes. And so they come together in fertilization to produce the zygote, which is the fertilized egg, which now have two copies of every gene, one from each parent. So every fertilization is a new combination of genes from both parents. When we inherit our chromosomes in meiosis, our genes are located on specific chromosomes. So we have 23 pairs, so we have 23 different chromosomes. We have about 75,000 genes. So those genes are scattered on those 23 chromosomes. So each chromosome has hundreds or thousands of genes. But it's not a random process. Genes are located at specific sites on a specific chromosome. So the position of a specific gene is called the gene, oops, that's too dark, the gene locus. Let me erase it. And so they know, like, if you have a certain genetic disorder, which gene which chromosome that is on and where that gene is located on a particular chromosome. Gene linkage is just referring to that some genes are very close together on the same chromosome, and so they're kind of connected. So this is a picture of, this here is a pair of sister chromatids joined at the centromere. And then this is a sketch, this one is chromosome 17. And so that's the banding pattern. But locations on the chromosome of what genes are located where. This is the BRCA1 gene. The BRCA1 gene is the gene associated with the increased risk of breast cancer. And it's on the 17th chromosome. This number here, 17, refers to which chromosome it's on, and then the letter refers to which arm. So this is the P arm and this is the Q arm. And so this gene is on chromosome 17 on the Q arm, and the 21 just refers to distance from the centromere. This is chromosome 11. This gene here is the defect that's found in sickle cell anemia. So that defect is on chromosome 11 on the P arm, and that's the position of it. So that's used to designate where specific genes are on specific chromosomes. So as we talked about during meiosis, the chromosomes are separated, and so you have 23 chromosomes in a gamete. And remember, the gametes are sperm and egg cell, so that the offspring that is formed after fertilization has one set of chromosomes from each parent. So this and those sets of chromosomes that are in the diploid cell, the 46 chromosomes, there's 46, which are 23 pairs. The pairs are your homologous chromosomes. So the homologous chromosomes have the same genes for the same traits, but they could have different versions of those genes. So this is, say, the chromosome that you got from one parent, and this is the chromosome you got from the other parent. And so they both have the gene that's indicated by a G. The capital versus lowercase level indicate different forms of that chromosome. So you could have gotten the same form from each parent, or you could have gotten different forms from each parent. The different forms of a gene are called alleles. So you can have two alleles that are the same or two alleles that are different. And so for every gene, except for those on the sex chromosomes, the X and Y, which we'll talk about separately, But on pairs 1 to 22, every gene, you have 2 alleles for that gene, one that you got from each parent. Two other terms, there's lots of terminology we'll be using as we talk about genetics. Two other terms that I'm going to talk about are genotype and then phenotype. So first, genotype. Genotype refers to which 2 alleles you got. So if your 2 alleles are identical, then you're homozygous. If you have 2 different alleles, then you're heterozygous. Zygos is the zygote. Homo means the same, hetero means different. And that's your genotype. So your genotype can either be homozygous or heterozygous. Phenotype is the appearance of that trait. So it could be like brown hair, blue eyes, green eyes, brown eyes. So that's the phenotype associated with a particular genotype. And we'll be talking about those. First, we're going to talk about, this is a karyotype. We looked at these on the last PowerPoint. So a karyotype is the picture of your chromosomes, and you have 23 pairs. Pairs 1 to 22, so up to this point here, are called autosomes. And autosomes, everybody has a homologous pair of the autosomes, unless you have some chromosomal abnormality, a monosomy or a trisomy, then you would have an abnormal number. But normal conditions, you have 2 of every chromosome for 1 to 22. And so those are called autosomal chromosomes, and both males and females have a homologous pair. The last pair, the 23rd pair, are your sex chromosomes, and those are different, and we're going to talk about them separately. But at the beginning of genetics, we're going to talk about autosomal genes that are on chromosomes 1 to 22. So if you have 2 of the same chromosomes, you're homozygous. And 2 of the same genes. We tend to use letter abbreviations for alleles. And the letter itself doesn't really matter that much. Sometimes it is based on the dominant phenotype, but we'll talk about that in a minute. So this is a parent that has 2 of the same allele, so they're homozygous. If we do this for eye color, they're homozygous for brown eyes. So during meiosis, where they're making haploid gametes, they can only give a big B to their gametes. So their sperm and egg cells could only have a big B. If they're homozygous for the little b, their sperm and egg cells could only get a little b. If they're heterozygous, their sperm or egg cells, 50% of them would get a big B, 50% would get a little b. For many traits, the big B and the little b indicate what's called dominance. And this is the example for eye color. Eye color is actually a little bit confusing, but it's the example everybody uses, so I'll use it too. And this only refers to brown, blue eyes. Green and hazel are variants of this. We're not going to really talk about them, but brown is dominant over blue, and so it gets the big B. Blue is recessive, so it gets a little b. And that's the way that the lettering is used. The dominant phenotype, that gets the big B, the recessive gets the lowercase level. In this case, because brown and blue both start with B, it's the same. It looks like you're just using the little letter, but suppose recessive was purple eyes, you would still use a little b because it's the lowercase of the dominant allele. So even if it wasn't blue, which starts with B, if it was purple was recessive, you would still use a little b. So a heterozygous has one brown allele and one blue allele. So half of their gametes would get the brown allele during meiosis and the other half would get the blue allele. If you only have two brown alleles, then you can only give your gametes a brown allele. If you only have blue alleles, you can only give a blue allele to your gametes because that's the only kind you have. So if you are homozygous, your alleles are the same as we talked about earlier. So if you're homozygous dominant, that means you have two brown alleles and you can only give gametes a brown allele. If you're homozygous recessive, that means you have blue eyes and you have two blue alleles, which are the little b's, and you can only give your offspring a little b. If you are heterozygous, that means you have one dominant allele and one recessive allele. So half of your gametes would get the brown allele and half would get the blue allele. So homozygous dominant has two dominant alleles. Homozygous recessive has two recessive alleles, and heterozygous has one of each. Now, a heterozygous, because brown is dominant, if you have a heterozygous genotype, so one big B and one little b, you have brown eyes because brown is dominant over blue. It's somewhat confusing because it sounds like brown is stronger or something. It just means that if the blue allele is a mutation and that mutation results in not any pigment being made in the eye, and with the absence of pigment, it's a blue color. But if you have at least one of the brown alleles, then you make enough brown pigment to have brown eyes. So the only way you can have blue eyes is to have two blue alleles. If you have either two brown alleles or one brown and one blue, you'll have brown eyes. So that individuals that are blue-eyed are homozygous recessive. So you know that they're homozygous recessive because they have the recessive phenotype, which is blue eyes. So they have to have two blue genotypes. Let me see. So that illustrates that an individual can have a different genotype, but the same phenotype. So big B, and big B, little b are two different genotypes, but they both are brown eyes. And brown eyes is the phenotype, so they both have the same phenotype. One of the tools that are used to predict outcomes, it's called from a cross, but since we're only talking about humans, that's really like combination of fertilization. And so when you have sperm and egg coming together with different alleles and genotypes, what would be the possible genotypes of the offspring? And the possibilities for each outcome are given by a Punnett square. So a Punnett square is a way of predicting possible outcomes. Doesn't mean that's the way it has to happen, but it means that that is the way, the likelihood. So it's kind of like tossing a coin, that if you toss a coin, you say you have a 50-50 chance of getting heads or tails, but that doesn't mean you can't toss it 4 times in a row and get heads each time. So it's only showing you the probability, which would be 50%, but it doesn't mean that that is what is actually going to happen all the time. So a Punnett square is used to show you possible outcomes of a particular sperm with a genotype and an egg with a particular genotype coming together to form an offspring. And so it's a tool for predicting outcomes. Hi, this is an example of a Punnett square, although you don't have to include the smiley faces. But with a Punnett square, you have 2 parents, which are indicated here. Sorry. Hi. Which are indicated here. So here's one parent who's heterozygous, has a big B and a little b, so has a brown eye allele and a blue allele. And the other parent, who is also heterozygous, big B, little b. So when you do the Punnett square, you draw a box. And then if you're just looking at one gene, you divide it into 4 sections, like that. And then you put the possible gametes from each parent along the side. So say this one is the mom. She can either give her offspring her big B or her little b. And this is the dad. He can either give big B or little b. Doesn't matter which side you put the male and the female on. And then what you do is just do the combination. So this box, the offspring would get the 2 big B's. In this one, they get this big B and this little b. And so what would this one be? A minute. Would also be a big B and a little b. And then the last one, this is just not working well, would be 2 little b's. So with this combination of parents who are both heterozygotes, you have the possibility of having a child with 2 brown alleles, a brown and a blue allele, and 2 blue alleles. And then the percentages are gotten from the boxes. So each box is 25%. So you have a 25% chance of having a child who is homozygous dominant, a 25% chance of having a child who is homozygous recessive, and then a 50% chance of having a child who is heterozygous. So that would be the genotypes. The phenotypes would be what their eye color is. So this one, and this one would all have brown eyes. So you have a 75% chance of brown eyes. And the only one that would have blue eyes is this one. So that would be a 25% chance of blue eyes. So that would be the predictions if you cross 2 heterozygotes for eye color. You would have a 75% chance of brown eyes, 25% chance of blue eyes. And that would be for any dominant recessive trait where both parents are heterozygotes. So they have one of each allele. You would see this outcome on your Punnett square. This is just using earlobe attachment. You can see the same kind of thing where you have 2 parents that are heterozygous for attached earlobes. Attached earlobes is kind of where you don't have a flap so much on your earlobes, and you would get the same possibility of a 25% chance of having attached earlobes. This is a practice Punnett square. So if you're doing this, you can pause the PowerPoint for a second and practice doing this Punnett square. So you have to figure out what are the possible genotypes and phenotypes of the offspring of a man who's heterozygous for eye color and a woman with blue eyes. So a man heterozygous, what would be his genotype? It would be big B, little b, right? And a woman with blue eyes would have to be little b, little b. So you want to do a cross with big B, little b, crossed with little b, little b. So take a minute and try doing that. I'm going to keep going with the PowerPoint because I run out of time with it eventually. So these would be your crosses. So here's the man who's heterozygote. Here's the female who's homozygous recessive. And so when you fill in your Punnett square, this would be big B, little b. This one would be little b, little b, big B, little b, little b, little b. So with this cross, where you had one parent who had blue eyes, you have a 50% chance of having a child who is homozygous recessive with blue eyes. And then the other 50% is heterozygous with brown eyes. You see, with this cross, there's no way you can get a big B, because this parent only has little b's. So all offspring are going to have at least one little b. This is just a collection of, what we're going to talk about next are disorders associated with a mutation in an autosomal gene. So again, those are on chromosomes 1 to 22. Both parents have one of those chromosomes. And there are some disorders, but not many, that are called autosomal dominant. So for an autosomal dominant disorder, if you have one copy of the dominant gene, then you have the disorder. So you could have big A, and you would have it. If you have a heterozygote and you have big A, little a, you would also have it because you only need one copy of the disorder. The only individuals who do not have it are little a's, little a's. There aren't as many autosomal dominant disorders because if they're serious disorders, they tend to disappear from the population, because if they're fatal, then individuals that have that gene aren't able to reproduce and pass it on. The reason that the recessive ones are in the population more is because you can be a carrier and not have the disorder. And that's in autosomal recessive. We're going to talk about those second, but first we're going to talk about autosomal dominant. So that you would have the disorder either if you had just one copy or two copies of the faulty. There aren't too many of these, as I mentioned. I'm going to just talk about two of them. One is Huntington's disease. And Huntington's disease is a defect of a gene on the 4th chromosome, the P arm. And this is considered to be a fatal disease. So again, you would think that it would disappear from the population if people inherit that trait. You can't be a carrier. You either have it or you don't have it. But the reason that it stays in the population a little bit longer is because symptoms don't usually appear till your 40s, 50s. So you might have it and reproduce and pass that gene off before you have any symptoms to know that you have the disorder. If it is present in your family, you can get a test done to determine if you have the faulty gene, and then you would know whether or not you are capable of passing it on to offspring. The other one that we'll talk about briefly is hypercholesterolemia. First, let's look at Huntington's disease. And Huntington's disease is a neurological disease. The defective gene is on the 4th chromosome. And again, symptoms usually don't start until 30s or 40s, or maybe even a little bit later. And you get progressive neurological deterioration with loss of being able to walk, talk, eventually even being able to feed yourself. There's severe dementia. So it's a pretty horrendous disorder. There's a link here for a video about a young woman who got the test done to see if she had that. Hopefully that will work if you click on it, but I'm not positive. Normally we would look at it in class. So this is a particularly horrendous disease because it causes such severe neurological abnormalities. And again, if you don't get symptoms till you're in your 40s and you didn't know that you had it, you could have already passed that gene off to your children before you knew that you had it. So that's Huntington's disease. It's autosomal dominant. The other one is hypercholesterolemia, which is a big word. Hyper means increased, emia means blood, and cholesterol. So this is very high blood cholesterol levels. Now, high blood cholesterol levels is very common, but this is a much more severe case that starts earlier in life than what would be the normal kind of high blood cholesterol levels you see in people as more related to diet as they get older. And so here the cholesterol is so high that it even gets like deposited. Here's like cholesterol deposits in the eyelid. There's some spots here in the cornea of the eye, or the iris of the eye, along the ankles. And then the more dangerous thing is that it also gets deposited in your arteries, where it can lead to heart attacks and stroke. So this also can be diagnosed, and there can be medication and stuff that is given to help control cholesterol levels. This is just terminology. I'm not going to go through. This is just kind of a list of all the things that we talked about. That is the end of the part 1 of the genetics presentation. The second part, we're going to talk about autosomal recessive disorders. But there will be a quiz associated with this part on campus. So see you soon. Bye.